Hello, this is microbiology. Today we'll be discussing somewhat about another basic topic that is indole test. So this is the main basic key point for today topic introduction. We discuss the principle, method, reagents, and result and interpretation, quality control, and summary of this test. So let's start the introduction of indole test. This test is basically demonstrate the ability of certain bacteria to decompose the amino acid tryptophan to indole. So it's mean there are certain bacteria they decompose the amino acid molecule tryptophan to indole. So indole, what is indole? Indole is basically the end product of the amino acid tryptophan. So tryptophan is basically decomposed and broken down by the bacteria, certain bacteria, and at last at the end product we have the indole. So this indole is basically accumulate in the medium, and indole production test is important in the identification of enterobacteria. So, what kind and what type of bacteria produce the indole, and what type of bacteria are indole positive? They are enterobacteria, most strain that is E. coli, P. vulgaris, P. retigeri, and morgani, and providentia species. So it's basically broken down the amino acid tryptophan and it release the indole in a culture medium, and they build up and accumulate in the culture medium. So what are the basic principle of indole test? That are when certain bacteria have a tryptophanase enzyme. So basically tryptophanase enzyme is the deaminase are broken down the tryptophan in a culture medium, and at last end product will be indole. So basically, we detect this indole by certain methods, and this uh, in this test. So where the tryptophan come from? Tryptophan is basically a requirement, a nutrient that are present in a culture media for the certain bacteria, and certain bacteria use this tryptophan molecule as a biomolecule for their growth. So how the bacteria produce indole? Actually, bacteria contain an enzyme tryptophanase. They basically catalyze the deamination, and which the amine group is removed from the tryptophan amino acid, and at last we have a final product remain that are indole. So how this indole molecule is detected in a culture medium? So this indole molecule is basically detected by two methods. We have quake reagent. We use in one method we use quake reagent, and the second method we use dimethyl aminoaldehyde, that are spot test method. So the first method is when indole is combined with quake reagents, which contain hydrochloric acid and p-dimethyl aminobenzyl aldehyde and amyl alcohol. So it's basically in a quake reagent we have HCl and p-dimethyl aminobenzyl aldehyde in amyl alcohol. The solution is basically turned from yellow to cherry red. So why amyl alcohol is used? Because that are not soluble in water, and the color that are produced in the tube will be the top of the broth. Within the tube there will be oily layer, and the color produced if indole is present, and the color is produced in the oily layer of the top of the broth. So we can easily interpret the result, and we can easily identify. So the second method of detection of indole is that the spot test. That when the indole combine in the filter paper matrix at an acidic pH with p-dimethylamino aldehyde DMACA to produce a blue to blue green compound. Indole spot reagent has been reported to be useful in detecting indole production by a member of family Enterobacteriaceae and certain anaerobic species. So on the basis of two methods, we detect the indole. We have to certain reagent. So which reagent is used in indole test is the so for the indole spot method we have a reagent is p-dimethyl amino aldehyde DMACA and hydrochloric acid deionized water. And the second one is the indole quake reagent method. Indole quake method. So what reagent are used in this method is the p-dimethyl amino benzyl aldehyde and second one is hydrochloric acid and amyl alcohol. So the hydrochloric acid is common between these two methods. So on the basis of these different reagent in these two method use, we have also production of different color compound. So by this different color compound we identify the indole. So let's discuss the method of to detect the indole test is one by one. So procedure of indole tube method that we take a sterile test tube containing 4 ml of tryptophan broth and inoculate the tube aseptically by taking the broth from 18 to 24-hour culture and incubate the tube at 37 degrees centigrade for 24 to 28 hours. Add 0.5 ml of aqueous reagents to the broth culture and observe for the presence or absence of a ring. So in a previous slide we discussed the tube method of the indole test. We have a circular ring is colorful ring, red color ring is produced on the top of the broth when there indole found in a culture medium. So the another important point is that why we take 24-hour culture because the tryptophanase enzyme that contain are present in a bacteria that are not stable for a prolonged period. So that's why we must take 24-hour culture. If we take more than 24-hour culture organism, so the test will be invalid. So result is in a positive result we have formation of a pink to red color that's called cherry red ring and the reagent layer on the top of the medium and that color will produce immediately within a second. And in case of negative we have no color change even after the addition of appropriate reagent. So this is the diagrammatic overview of the indole test is look like. We have indole positive. The top of the broth we have a cherry red color appear, while when indole negative we have no any color on the top of the broth. The second method for identifying the indole molecule is the indole spot test. Place several drops of 1% P dimethyl amino quinone amaldehyde reagent on a piece of filter paper until saturation. With inoculating loop or wooden applicator stick, we pick a portion of an 18 to 24 hour isolated colony from a non-selective media and wrap it onto the reagent saturated area of the filter paper. So simply we take the colony from the culture up 18 to 24-hour culture, and we rub this colony on a filter paper. So look for observing the color development within 1 to 3 minutes. So in a quake method, we interpret the result within a second, or we interpret the result immediately. While here in an indole spot test, we observe the color development within 1 to 3 minutes. So in case of a positive indole reaction is denoted by the appearance of blue to blue-green color change on a bacterial smear within 2 to 3 minutes. In case of a negative result, so negative reaction will be remain colorless or light pink. Another important point is that when positive reaction is red violet in case of a Providencia alkalipians. So on a basis of different method, we have also a different reagent in use in these two methods. So the color will also be different on the basis of these different reagents. So basically, this is the diagrammatic overview of spot test, indole spot test. So the first we take indole spot reagents on a filter paper. Then we impregnated the colony or culture on a these spot reagent that are present on the filter paper. If we found any color on a filter paper, the test will be positive. If we not found any color, the test will be negative. Quality control. So in the indole test, we use positive control, and the second one is negative control. So positive control is the E. coli, that is NCTC 10418, and negative control is Proteus mirabilis NCTC 10975. So the control is basically used for every single test, because we by this control we actually assure and assure our result. So E. coli, why E. coli we take as a positive control? Because E. coli itself, they are indole positive bacteria. So that is why we take this bacteria as a positive control to validate our result according to the result positivity. And why we take Proteus mirabilis as a negative control? Because they are indole negative. So by this negative control, we validate our result regarding as a negative test. So at last, the summary of the topic is that the indole test is basically performed to differentiate certain bacterial species from each other. Certain bacteria are indole positive, while certain bacteria that are indole negative. Proteus mirabilis, indole negative bacteria, that will be different from all other Proteus species. This is indole positive. So basically, Proteus mirabilis, that are indole negative bacteria, while the rest of other Proteus species that are indole positive. To differentiate between the Klebsiella pneumonia, that are indole negative, from Klebsiella oxytoca, that are indole positive. To differentiate between the Citrobacter porondi, that are indole negative, while Citrobacter coceri, that are indole positive. Thank you for watching.